Welcome back to the deep dive. We have a really interesting setup on the desk today. Usually when we tackle a subject, our sources tend to agree on the general premise. Everyone is usually rowing in the same direction.
It is a stark contrast today, isn't it? It's pretty rare we get friction like this right out of the gate.
It really is. Because on one hand, we have this field guide from the Pacific Northwest of North America. It's vibrant, it's enthusiastic. I mean, it's essentially a menu.
Right, it paints this picture of a fungal paradise where the woods are just overflowing with food.
Yeah, it's telling you how to find these massive, abundant mushrooms and basically fry them up in butter. It feels like a treasure hunt.
But then sitting right next to it, we have this completely sobering scientific paper from Belarus in Eastern Europe.
Exactly. And it's not a field guide at all. It's an analysis of the Red Data Book. It's a list of species that are on the brink of extinction, protected by law.
And the kicker, the part that really makes you stop and think, is that they are talking about the exact same mushrooms.
The exact same one. So our mission today is to bridge that gap. We are talking about toothed fungi.
Also known as hydnoid fungi.
Right, hydnoid fungi. We're going to figure out why one forest dinner is another forest's endangered species. But before we get into the ecology and the politics of forest management, we have to start with the biology.
Because we have to clarify what we mean.
Exactly, because toothed fungi is a bit of a misnomer, isn't it?
It is entirely a linguistic trap. When you hear the phrase toothed fungi, your brain naturally wants to categorize it like a family tree.
Like they're all related.
Exactly. You think, okay, these are the Smiths and those are the Joneses. You assume they share a recent genetic ancestor. But biologically, that is completely wrong. Toothed isn't a family.
It's a job description.
It's a shape. Specifically, it's a morphological solution to a very basic physics problem.
Right, because if you are a mushroom, your primary biological imperative, your one true purpose in life, is to drop spores. You need to get those genetic packets out of your body and into the wind currents.
And you could theoretically do that on a flat surface, just a flat patch of tissue.
But that's terribly inefficient. You'd run out of room very quickly.
So you need to maximize surface area. You need to pack way more spore-producing real estate into the exact same amount of spatial volume.
Precisely. And evolution has solved this geometry problem in a few distinct ways. You can fold the surface into plates. Those are the gills you see on a standard button mushroom or portobello.
Or you can punch little holes in the surface. Those are pores, like you see on a sponge or a bolete mushroom.
Or, and this is what we're looking at today, you can grow long, hanging spines. Icicles, essentially. And you cover every single millimeter of those hanging spines with spore-producing cells.
That is the toothed strategy. And it's brilliant because gravity helps the spores fall off the spines, clear the mushroom body, and then the wind just takes them away.
And what's absolutely fascinating is that nature didn't just invent this mechanism once. It wasn't one genius mushroom eons ago that came up with this design.
No, and this is the first big aha moment for this deep dive. This is a classic case of convergent evolution.
Right. You have completely unrelated genetic lineages, groups of fungi that haven't shared a common ancestor for millions of years. And they all independently arrived at the tooth design just because it works so well.
It's exactly like how birds, bats, and insects all invented wings completely separately. The environment demanded a solution, and multiple unrelated groups found the same one.
The notes mention a specific mushroom that really hammers this point home: the toothed jelly.
Oh, yes.
Pseudohydnum gelatinosum. Even the scientific name sounds like it's actively trying to trick you.
Pseudohydnum literally translates to false hydnum, and it is the perfect example because if you look at it under a microscope or if you analyze its DNA, it is unequivocally a jelly fungus.
Right. It's related to those shapeless, gooey blobs like witch's butter that you see shivering on rotting logs after a rainstorm.
Exactly. It is not related to the other mushrooms we are discussing today at all, but somewhere along its evolutionary line, this specific jelly fungus hit an evolutionary bottleneck where it needed better spore dispersal. So it grew teeth.
But it kept the jelly texture. It didn't turn woody or fleshy like a normal mushroom.
Oh, absolutely not. The description in the Pacific Northwest Field Guide is just fantastic. It's described as flabby, rubbery, gelatinous.
That's quite the hyphenation.
It really is. It's this translucent, whitish-gray thing, usually shaped like a little tongue or a fan, just sticking horizontally right out of the wood.
And the teeth themselves?
If you touch them, they feel weirdly soft. The guide compares it to a cat's tongue or a wet gummy bear.
So it is literally a jelly fungus wearing a toothed fungus costume.
That's exactly what it is. It proves that teeth are just a functional interface. It's a successful biological strategy that nature keeps 3D printing across entirely different fungal lineages.
Okay, so we've established that we are looking at a diverse group of strangers who all happen to shop at the same spiny clothing store. Let's organize them a bit for the listener.
That's a good idea.
If you walk into the woods right now, what are the main body types of these fungi that you should be looking for?
We can break them down into 3 distinct architectural styles. The first is what mycologists call stipitate.
And stipe is just the scientific word for a stem.
Correct. This is the most familiar form to anyone who has ever drawn a picture of a mushroom as a kid. These look like your standard terrestrial mushroom. You have a cap on top, a stem below, but instead of gills underneath the cap, you have a dense bed of hanging spines.
This covers general like Hydnum and Phellodon. Easy enough.
Fun.
Cap, stem, teeth.
Right. Then we had the weird ones, the framework style.
These are the showstoppers.
They really are. Think of a coral reef or maybe a frozen fountain. They do not have a cap at all. They branch out like a complex, intricate coral structure, and the branches themselves are entirely covered in hanging spines.
This is the Hericium genus, which we'll spend some real time on because visually they are just stunning.
And then there's the third type, the one that doesn't look like a mushroom at all to the untrained eye.
The resupinate fungi.
Exactly. The term comes from Latin, meaning lying on the back. These don't have a stem, and they don't have a cap.
They just look like a splash of paint.
A splash of paint or a hard crust growing completely flat against the underside of a rotting log. But if you look closely, if you get right up to the wood, that crust is covered in thousands of tiny teeth projecting outwards toward the ground.
These are the ones people usually ignore. You see a crust on a log and you just think it's generic rot.
They do get ignored.
Okay.
But as we'll see when we get into the Belarus data, these crusts are actually some of the most critical indicators of an ancient, healthy forest ecosystem. They are the hidden gems of the mycological world.
Let's start with the celebrities, though. The framework fungi. The genus Hericium. If you go on social media or look at foraging forums, especially in the Pacific Northwest, these are the absolute superstars. You see people holding these massive white things that look like pom-poms or cascading waterfalls.
They are incredibly charismatic. You really can't mistake them for anything else in the woods. They look like waterfalls of white icicles suspended in midair.
Our Pacific Northwest source breaks down 4 specific species within this genus. And I want to clarify the differences here because I feel like people tend to lump them all together under the common name lion's mane. But there are important structural nuances.
That happens a lot in foraging communities. Lion's mane properly refers to just one specific species, Hericium erinaceus. Morphologically, it's distinct from the others because it is an unbranched, completely solid mass.
So no coral-like arms.
None. It looks like a single large snowball, or maybe a bearded face, entirely covered in long, cascading spines. And these spines can be huge, up to 5 centimeters long.
Okay, so if it's branching out like a piece of coral, it is absolutely not lion's mane.
Correct. If it's branched, you're looking at one of the cousin species. Take Hericium coralloids, for instance, the comb Hericium. This one is highly intricate. The spines hang down from the branches in very neat, ordered rows.
Exactly like the teeth on a plastic hair comb.
Precisely. It's very delicate, almost lace-like in its structure compared to the dense snowball of erinaceus.
And then we have the giant of the group, Hericium abitus.
The conifer coral. The source mentions mature specimens reaching up to 75 centimeters across.
That is a mushroom the size of a beach ball.
Imagine stumbling across that in a dark, damp, old-growth forest, a glowing white beach ball of fungus just hanging off a dead tree.
It's massive.
Yeah.
And the scientific name, abiatus, gives away its location, right? Abies is the genus for fir trees.
Exactly. This particular species is strictly a conifer lover. If you are hiking in a fir or hemlock forest in the Pacific Northwest and you see a giant coral-like fungus, it is almost certainly abiatus.
But if you find a similar-looking framework, coral fungus on a deciduous hardwood tree, like an oak or a maple, it's something else.
Right. Then it's likely Hericium americanum. Host tree identification is actually your best field tool for distinguishing these species. The mushroom itself tells you what kind of tree it is currently eating.
Now, this is exactly where the 2 source documents collide in a really fascinating way. In the U.S., finding a Hericium is a major score. You take a picture, you harvest it, you take it home for dinner. It's a great day in the woods.
It's a prize.
But then you flip over to the Belarus paper, and it lists Hericium coralloids as Category 2. Endangered.
A completely different reality.
Why the drastic difference? Why is it a feast in Oregon and a strictly protected species in Belarus?
It all comes down to habitat continuity and the forestry definition of what makes a clean forest. Hericium species are what ecologists call indicator species.
Meaning they tell us something about the broader health of the environment.
Yes, they are notoriously picky. They don't just grow on any random piece of deadwood. They require large diameter dead trunks, what foresters call coarse woody debris. And, crucially, they need forests that haven't been subjected to sanitary cutting.
Sanitary cutting. The Belarus paper lists this as a primary existential threat. To a layman, sanitary cutting sounds like a positive thing. Hygiene for the forest. Keeping things neat.
It sounds totally benign, doesn't it? Like brushing your teeth to prevent cavities. But ecologically speaking, it is disastrous for these specific fungi. In heavily managed forests, especially across parts of Europe, there is a very long tradition of systematically removing dead standing tree snags and pulling out fallen logs.
To prevent pine beetle outbreaks, or sometimes just to tidy up the woods and harvest leftover firewood.
Exactly. But Hericium is a white rot fungus. Its entire biological existence depends entirely on having decades to slowly digest that large deadwood.
So when forest management comes in and cleans the forest, they are literally evicting the fungus.
You're removing their housing and their food source simultaneously. You are bulldozing their entire neighborhood. The Pacific Northwest still has vast tracts of old-growth or mature secondary forests with enormous biomass. Giant logs just sitting there, rotting on the damp ground for 50, 60 years.
That's why Hericium is still abundant there. There's plenty of food.
Yes. Whereas Belarus and much of Eastern Europe has seen much more intensive fragmented forest management over several centuries, it's a stark lesson in how messy forests absolutely full of chaotic, rotting, dead stuff, are actually the healthiest forests for biodiversity.
That's a critical takeaway for anyone interested in conservation. If a forest looks too tidy, it's probably a biological desert.
A tree farm is not a forest.
Well said. Okay, let's move from the wood rotters up in the trees down to the ground dwellers. Let's talk about the other big edible favorite from the guides, the hedgehogs. The genus Hydnum.
These are often referred to as the safe edibles. If you are a beginner forager, this is exactly where you want to start your journey. They are terrestrial, meaning they grow right in the soil and leaf litter, not erupting out of wood. They have a classic mushroom cap, usually a pale orange or cream color, and underneath that cap you find these brittle little teeth.
Why are they considered so safe? Is it simply because they don't have poisonous twins?
That is exactly why. They have absolutely no poisonous lookalikes. If you find a mushroom with teeth that grows on the ground, the worst that happens if you misidentify the exact species is that it tastes a little bitter or has a tough, corky texture. It won't put you in the hospital. That's a very comforting fact when you're just learning field identification.
The Pacific Northwest Field Guide splits the hedgehogs into two very broad, easy-to-understand groups. The big ones and the little ones.
Roughly, yes. That's historically how we categorize them in the field. You have the Hydnum repandum group. These are the large, chunky, pale, orange to buff-colored ones. A key morphological feature here is that the teeth are decurrent.
Meaning the teeth don't just stop at the edge of the stem. They actually run down the length of the stem a little bit.
Exactly. They trail down the stipe.
And the little ones.
Those belong to the Hydnum umbilicatum group. These are physically smaller, usually a much deeper, richer reddish-orange color. And the cap has a distinct belly button, a little prominent depression right in the dead center. That's actually what umbilicatum means. Navel.
But of course, science can't leave things that simple and easy for the foragers. The notes bring up a really interesting 2018 molecular study by a researcher named Niskanen.
Yes, modern DNA analysis has completely shattered that simple big versus little binary. It turns out that what we confidently thought were just two widespread species are actually a complex of many different genetically distinct species. In the Pacific Northwest alone, this study identified at least 6 completely distinct species within those two groups.
Giving them new localized names like Hydnum olympicum and Hydnum oregonensis.
Right. They look virtually identical to the naked eye, but their DNA proves they are reproductively isolated lineages.
So does this molecular splitting actually matter for the casual forager?
For the cooking pot, no, not at all. They all taste identical when sautéed. But for conservation ecology, It matters immensely.
Because it completely changes the population math.
Exactly. It means that instead of having one giant, robust population of Hydnum pandem spanning the entire continent, we might actually have incredibly small, localized, fragmented populations of things like Hydnum olympicum. It fundamentally changes how we assess extinction risk. If a newly discovered species only lives in one specific river valley, it's exponentially more vulnerable to logging or climate change than we previously realized.
It's the classic splitter versus lumper debate in biology, but with real-world survival stakes.
Precisely.
Let's step out of the kitchen now and put on our lab coats. We are moving into part 3 of our deep dive, the Mycorrhizae family, often just referred to affectionately as the Leatheries.
Or the cork fungi. These are incredibly tough, densely fibrous, and generally entirely inedible due to their texture. You would never want to try eating one of these. It would literally be like trying to chew on a hiking boot. But they are fascinating because interacting with them is a multi-sensory experience.
Let's talk about the smell, because this is wild. The genus Phellodon.
Phellodon is famous in mycological circles for its distinct odor, especially as the fruiting body dries out. The field guide describes the scent as fenugreek, sweet maple syrup, or even Maggi seasoning.
Maggi seasoning. For anyone who hasn't used it, that is a very specific, intense umami celery salt soup flavoring. That is a radically specific scent to find in the middle of a forest.
It is. It's a spicy, savory, almost curry-like aroma. Imagine you're walking through a damp, cold pine forest, and suddenly you catch a strong whiff of pancakes or a curry house. You look down at your boots, and there are these dark, tough, funnel-shaped mushrooms pushing up through the pine needles.
And their growth habit is weird, too. The notes use the phrase indeterminate growth. What exactly does that mean in this context?
Most fleshy mushrooms, like a standard button mushroom or a portobello, grow to a specific, genetically predetermined shape, and then they stop. They have a biological architectural plan. Phellodon and its close cousins just keep going.
They just continuously expand.
Yes. If it rains, the edges expand outward. If the weather turns dry, they simply pause their growth and wait. Because of this stop-and-start expansion, they end up growing entirely around obstacles. You'll find them actively swallowing fallen twigs, incorporating pine needles into their caps, and even engulfing neighboring mushrooms.
They just slowly swallow the forest floor. That's a bit unsettling.
They fuse together into these massive, multi-capped, convoluted masses.
Now, for the hardcore identification nerds listening, there's a specific challenge with this group, the black Phellodons. Specifically, differentiating Phellodon atratus from Phellodon melaleucus. They look completely identical.
To the naked eye, yes. Both are dark, blackish-gray, tough, and leathery. If you hold them side by side, you cannot tell them apart visually. This is exactly where you need your field chemistry kit. Specifically, a little dropper bottle of KOH potassium hydroxide.
You literally drop a harsh chemical on the mushroom right there in the dirt.
Right there in the woods or back at your kitchen table lab. If you slice open the flesh of Phellodon atratus and apply a single drop of KOH, the flesh instantly reacts and turns a deep, inky blue-black. But if you do the exact same thing to Phellodon melaleucus, the flesh reacts by turning a dark, murky olive green.
That's a really subtle distinction. Blue-black versus olive-green.
It is subtle, but it's binary. It definitively tells you that they are chemically distinct species, even if their outward morphology is an identical clone.
And speaking of internal chemistry, we absolutely have to talk about the dyes.
Yeah.
The genus Archidon.
Ah, the hawkwings. These are big, highly impressive fungi.
Fleshy, with these massive overlapping scales on the cap that literally look like the ruffled feathers of a hawk's wing.
They are beautiful. And for a very long time, classical taxonomists genuinely thought Sarcodon imbricatus, which typically grows under spruce trees, and Sarcodon squamosus, which grows under pine trees, were the exact same organism. They look identical under a hand lens.
But the textile artists knew better.
The dyers were way, way ahead of the scientific establishment on this one. This is one of my favorite pieces of ethnomycology. Traditional wool dyers knew that if you took the pine-associating mushroom squamosus and boiled it in a pot with wool yarn, you yielded a stunning, vibrant, bluish-green color.
But if you used the spruce-associating one...
If you used imbricatus, you just got a dull, boring, grayish beige.
That is incredible. The specific tree partner that the mushroom connects to fundamentally changes the internal chemical payload of the fungus enough to completely alter the resulting dye color.
It perfectly illustrates that a species isn't just defined by its physical shape, it's defined by its complex chemical interactions with its specific environment. The dyers were effectively doing chemical taxonomy with their dye pots centuries before mycologists were doing it with DNA sequencers.
I want to highlight one more species in this tough and leathery group because it is just so visually startling. The bleeding tooth fungus, Hydnellum peckii.
The absolute goth fungus of the forest. It is a completely unforgettable sight.
Describe it for someone who hasn't had the pleasure of stumbling across one.
Imagine a lumpy, amorphous white cushion sitting directly on the forest floor, usually nestled in deep green moss, and across its pale surface it is actively oozing thick, bright, cherry red droplets. It looks exactly like a practical effect from a horror movie or maybe a jelly donut that someone just stepped on.
Is it actually blood? Or something akin to blood?
No, not at all. It's a physiological process called guttation. The fungus is rapidly metabolizing and expelling excess water from its internal structure. But as it pushes this water out, it carries along a deeply pigmented secondary metabolite. It's visually startling. And I should add, despite looking exactly like sweet strawberry jam or cherry candy, it is excruciatingly, painfully bitter.
So, standard foraging rule applies. Do not taste the bleeding tooth fungus.
Definitely not, unless you want your mouth to pucker for an hour.
Good tip. Okay, we've covered the tasty edibles, the smelly, leathery ones, and the bleeding ones. Now, I want to take the listeners entirely into the deep end. We are looking squarely at the Belarus Red Data Book's rare and resuppinate list.
This is usually the exact moment where the casual weekend hikers tune out, but I promise you, this is where the real complex ecology happens. We are talking about the crusts.
Resuppinate fungi. These are the ones we mentioned earlier that just look like a flat coating of paint on the underside of a log. Why are we so worried about a crust? Let's take Dentapellus fragilis, for example.
Dentapellus fragilis is essentially a ghost of the ancient forest. The specific scientific epithet, fragilis, refers directly to its teeth. They are long and stark white, but they are so unbelievably soft that if you brush your finger against them when they're dry, they simply disintegrate and crumble to fine dust. It's an incredibly delicate organism.
And it is notoriously picky about its home.
Right? Extremely. It strictly requires what we call naked wood of deciduous trees, specifically maple, alder, or hornbeam. And by naked, we mean a very specific stage of decay where the bark has naturally sloughed off, but the structural wood underneath hasn't yet turned to soft mush. It occupies a highly specific, narrow temporal window in the multi-decade rotting process of a tree.
The Belarus conservation paper specifically calls it an indicator of forests minimally affected by humans.
Right. Because in any commercially managed forest, a fallen tree log is almost never allowed to sit there completely undisturbed long enough to lose its bark naturally and reach that perfect stage of decay without being hauled away for pulp or crushed under the treads of heavy logging machinery. If you actually find Dentapellus fruiting, you are standing in a forest that has been left entirely alone for a very long time. It signifies an unbroken, ancient continuity of deadwood.
It's living history. Then there's Cythotrema raduloides. And this one is fascinating because it requires a microscope to even confidently confirm you found it.
Yes. You might find the white crust on a log, but to know for sure what it is, you have to look closely at the basidia.
The basidia are the microscopic structures that physically produce the spores, right?
Exactly. In almost all basidiomycete mushrooms, the basidia are shaped like little bowling pins or clubs, but in the genus Cystotrema, they are urniform.
Urniform, meaning shapes like an urn.
Exactly like a classical Greek urn. They have a swollen, rounded base that sharply constricts into a narrow neck before widening slightly at the top.
So you carefully scrape a tiny bit of this completely unremarkable white crust off a rotting log, you put it on a glass slide under the lens, and suddenly you are looking at a microscopic landscape of thousands of tiny Greek vases.
That's exactly it. And that specific geometric shape is the definitive diagnostic signature for the species. It's a level of intricate beauty that you simply cannot see with the naked eye. It forces you to slow down and zoom in.
The source material also briefly highlights Phlebia albumelli.
This one is interesting from a developmental morphology standpoint because it actively blurs the established lines. It starts its life cycle fruiting as tiny, distinct, cup-like bodies like minuscule little bowls scattered on the wood. But as they grow and mature, these individual bowls expand and merge together to form one continuous toothed crust.
It shows that the rigid taxonomic line we draw between a cup fungus and a crust fungus is actually quite fluid in nature.
Life doesn't care about our neat little categories.
This leads us perfectly to the broader question of ecology. What are these bizarre organisms actually doing? We know they look amazing under a microscope, but what is their actual function in the grand machine of the forest ecosystem?
They are essentially the engine room of the global carbon cycle. We're looking at two primary ecological roles here. First, we have the recyclers, the white rot fungi. This group includes the massive Hericium species and the delicate Dentapellis we just discussed.
Why do we call it white rot? Is it just because the fungus itself is white?
No, it's named for the condition of the wood they leave behind after they eat. Wood is primarily constructed of two things: cellulose, which is white and somewhat flexible, and lignin, which is brown, rigid, and incredibly hard. Lignin is the tough biological glue that physically allows a tree to stand upright against gravity and wind. It is an incredibly complex polymer, very difficult to break down chemically. White rot fungi are some of the only organisms on planet Earth that have evolved the specific enzymatic tools to digest lignin.
So they're essentially eating the brown glue.
They eat the brown lignin glue holding the wood fibers together, and they leave the stringy white cellulose behind. If you ever walk in the woods and pull apart a rotting log and the inside falls apart into these wet, white, stringy fibers, you are looking directly at the aftermath of their work. Without white rot fungi, dead trees would simply stack up indefinitely. We would be buried hundreds of feet deep in undecomposed wood, and all that vital carbon would be locked away forever.
So they literally keep the global carbon cycle moving. They are the great recyclers. And what about the second ecological role?
The second group are the partners, the mycorrhizal fungi. This includes the Belarus family, the Bankera, the Smelly Phellodon, and the scaly Sarcodon we talked about.
These are the ones that are physically connecting to the roots of living trees.
Yes, it's an intricate, symbiotic trade agreement. The fungus extends its vast underground web of mycelium deep into the surrounding soil. Because its threads are microscopic, it can mine the tiny crevices of soil and rock for vital minerals like nitrogen and phosphorus elements that the massive tree roots have a very hard time extracting on their own. The fungus pipes these dissolved nutrients directly into the root system of the host tree. In exchange, the tree performs photosynthesis up in the canopy and creates liquid sugars, which it pumps down into the roots to feed the fungus.
It's an economy. It's the internet of the forest, a massive resource exchange network.
It is a beautiful system. But this deep interdependence makes them doubly vulnerable to environmental changes. They are acutely sensitive to both the health of the host tree canopy and the chemical quality of the soil. The Belarus conservation paper specifically points out that these mycorrhizal toothed species are incredibly sensitive to soil acidification and nitrogen pollution.
When you say nitrogen pollution, you mean things like acid rain or agricultural fertilizer runoff from nearby farms.
Exactly. If the ambient nitrogen levels in the forest soil get artificially too high from human pollution, the tree suddenly doesn't need to trade with the fungus anymore. It can get the nitrogen for free. Or, alternatively, the soil chemistry simply becomes toxic to the delicate mycelium. Either way, these partner fungi just stop fruiting, retreat, and eventually die out completely. They serve as the ultimate canaries in the coal mine for hidden soil health.
So let's comprehensively look at the major threats outlined in the text. The Belarus paper explicitly lists intensive forest exploitation as the universal overarching threat. We already talked about the removal of deadwood through sanitary cutting, but the paper also specifically highlights the draining of swamp forests.
That has been a massive historical issue in Eastern Europe. Many of these rare-toothed species, like Dentapellis, absolutely rely on high, stable, ambient humidity. They evolved in dark, constantly damp, swampy, old-growth forests. But in many parts of the managed world, there is a long, profitable history of deliberately draining these wetlands. You dig deep ditches, you drastically lower the regional groundwater table, and you plant fast-growing timber pines.
And as a result, the ambient humidity drops. The forest dries out.
The forest fundamentally dries out, and the highly specific, humid microclimate that physically supports these fragile crust fungi simply evaporates into thin air. Their delicate fruiting bodies can't survive the new, prolonged dry spells.
And then there's the recreation effect. And that one points the finger directly at us. That's the casual hikers and nature lovers.
Unfortunately, yes. It is us walking off trail, trampling. And specifically, trampling and compressing the delicate moss layer. The terrestrial fungi, like Bankera and Phellodon, often rely heavily on a pristine, undisturbed carpet of moss to aggressively regulate moisture and temperature for their shallow mycelial networks. If a group of hikers walks through there and crushes that fluffy moss down into hard, compacted mud, you are effectively destroying the insulated roof over the fungus's head. You instantly alter the hydrology and aeration of the topsoil.
It really fundamentally changes how you look at a casual walk in the woods. You realize that the ground beneath your boots isn't just inert dirt, it's a living, breathing architecture.
It is entirely alive, and we really need to learn to tread lightly.
Before we move to wrap things up, I want to circle back to the hard science for just 1 minute, because the text goes deep here. We touched on field identification chemistry earlier with the KOH turning the Phellodon blue-black, but the notes heavily emphasize another common chemical test, Meltzer's reagent.
Ah, yes. Meltzer's reagent is an absolute staple in any serious mycologist's toolkit. It's essentially an iodine-based liquid solution, and its primary diagnostic use is to test fungal spores for the presence of specific starch-like compounds.
Like the classic iodine test we all did in high school biology, where you drop it on a slice of potato and it turns black.
Exactly the same underlying chemical principle. If the microscopic fungal spores contain these specific complex starches in their cell walls, they will instantly turn a dark, striking blue-black when the iodine solution hits them. Mycologists term this positive reaction amyloid.
Why does this specific microscopic reaction matter so much for classifying tooth fungi?
Because it serves as a definitive binary toggle for evolutionary identification. Remember how we said teeth were invented multiple times by unrelated mushrooms? Meltzer's reagent proves it chemically. The spores of the Hericium lineage are strongly amyloid. They turn black or deep blue in iodine. But the spores of the Hydnum lineage, the terrestrial hedgehogs, are inamyloid. They just stay clear or faintly yellowish.
So if you have a mystery spine-covered fungus and its shape is confusing, you can bypass the morphology entirely.
You just put the spores on a slide, drop some iodine on them, and look. Blue equals the Hericium evolutionary lineage. Clear equals the Hydnum lineage. It instantly cuts through the visual confusion of convergent evolution.
It's microscopic detective work.
It truly is, and it reveals that what looks confusingly similar on the outside, a hanging spine, is built from entirely different genetic instruction manuals on the inside.
And there's one last, incredibly specific microscopic detail mentioned in the text. Glueocystidia. That is a fun word to say.
It is quite a mouthful. Glueocystidia. These are highly specialized sterile cells, meaning they aren't the basidia that produce the actual spores. They are embedded among the spore-producing cells on the teeth, and they are packed full of dense, oily, highly refractive chemical contents.
So under a microscope lens, how do they look?
Because of the oil, they actively refract light. They look shiny, highly contrasted, almost glowing against the background tissue. Denapellus fragilis, for example, is absolutely packed with them. We don't always fully understand their exact biological purpose, maybe chemical defense, maybe moisture retention, but their presence is just another hidden, beautiful layer of evolutionary complexity that you'd miss if you didn't look close enough.
So let's take a step back and bring this massive amount of information all together. On one side of the desk, we have the sheer abundance of the Pacific Northwest, the enthusiastic menu of giant edibles. And on the other side, we have the fragile, hanging by a thread reality of the Belarus Red List. But when you look at the ecology, they're really just two sides of the exact same coin, aren't they?
They absolutely are. The Pacific Northwest, with its massive dead logs and deep continuity, is essentially what a red data book forest looked like centuries before it was threatened. It shows us the biological potential. It vividly shows us what an ecosystem is capable of when it retains ancient wood and deep, undisturbed moisture.
And Belarus.
Belarus shows us the inevitable consequence of losing that ancient continuity. It shows us exactly how fragile these complex systems are and how incredibly hard we have to work to save the fragmented remnants that are left.
It really just emphasizes the vital importance of looking closely at the world around us. Whether you are holding a dried Phellodon to your nose to catch that weird, surprising whiff of maple syrup, or meticulously checking the top of a Hydnum cap for that little umbilical belly button, or just finally realizing that a boring black crust on a rotting log isn't just mud and dirt.
It's about fundamentally realizing that the forest isn't a single green background. It's an intricate tapestry full of these highly distinct, ancient entities, each with a specific job.
So what is the ultimate takeaway for the listener today? Next time they are out there on a hike in the woods, what should they be doing?
Next time you see any mushroom, don't just look at the top of the cap and keep walking. Bend down, look underneath, gently use a mirror if you have to. If you see teeth hanging down instead of normal gills, I want you to stop for a moment. Realize that you are looking at an ancient biological lineage that only survived by mathematically maximizing its surface area in the most clever, gravity-assisted way possible. You are witnessing convergent evolution in real-time action.
And critically, respect the deadwood.
Absolutely. Please don't indiscriminately kick apart a rotting stump just for fun. That stump isn't garbage, it is a thriving metropolis. It is a complex city providing housing and food for Hericium, for Denapellis, for Cystotrema. In heavily managed landscapes, that singular piece of ugly deadwood is literally the final life raft for these endangered species.
I want to leave everyone listening with a final, somewhat provocative thought. We talk extensively today about how DNA analysis is rapidly changing the map. It's forcibly merging some species like Sarcodon, shifting over into the Hydnellum genus, and it's violently splitting others apart, like realizing the common hedgehog mushroom is actually 6 completely distinct species in one region.
Yes, the rigid classical concept of what defines a biological species is getting incredibly blurry the closer we look.
It heavily suggests that what we perceive with our naked human eyes, the outward morphology, the shape of the cap, the color of the spines, is only a tiny, often misleading fraction of the true biological story.
It really does raise a profound question. How many other recognized species in our field guides, things we think we perfectly understand, are actually just one highly adaptable shape-shifting organism drastically altering its body to respond to local environmental pressures?
Or conversely, how many things that look identical to us, 2 mushrooms sitting side by side on a log, are actually separated by tens of millions of years of invisible evolutionary history?
We truly are walking through a natural world built on visual illusions, and the only way to ever see the biological truth is to continually ask questions and keep diving deeper.
And maybe remember to carry a little bottle of iodine in your backpack.
It definitely couldn't hurt.
Thanks for joining us on the Deep Dive. We'll see you next time.
Keep looking down.